Hello, welcome to 13 Universal Learning Beyond Barriers. And this is Shedro, your biology tutor. So we are going to be looking at biological molecules, part one. So we're going to be discussing testing for biological molecule. This is a subject-specific content. Okay, so we have questions, all right, on this topic on this same channel. So now, if you have not watched other videos, maybe part two, part three on biological molecules, do watch them. But here we're going to be looking at the practical aspect of biological molecule, that is testing for the presence of biological molecules. Now, biological molecules, what are they? They are building blocks of life. Okay, this is a cell. This cell is made up of this biological molecule. For example, this glucose, of course. If you look at this, this looks like a protein. That's triglyceride. This is DNA. Wow, that stores a lot of information. Okay, this also looks like of course like a carbohydrate. This is an amino acid. Is that okay? So these are structures. So biological molecules are the molecules of life. All right, they are made up of monomers. The monomers join by a process called polymerization. Okay, to form biological molecules. So there are four of them. All right, five. We have water, we have carbohydrates, we have nucleic acid, protein, and then we have lipids. Is that okay? So we're going to check how do we know their presence, right? If it is in your food, how do you check whether the manufacturer's claim is correct? So look at this. The first one we're going to do is Benedict test for reducing sugars. So Benedict test is used to check whether reducing sugars, okay, are present in your food. Don't forget that there are three things that you will have to check out for. In tests for biological molecules, you have to look out for the reagent. So what reagent are we is involved, all right, in the test for this molecule? All right, so what chemical do we need to identify the presence of this molecule? That's number one. Number two, you have to look out for the procedure. What are the steps? You must know the steps, okay, that can be used to identify the presence of biological molecules. Is that okay? So procedures are very important. All right, yeah, and then we must look out for results. Okay, what color indications shows us that the biological molecule is present or absent? Is that okay? So guys, let's do this. So watch this. So the first thing we'll look at here is Benedict solution. Now Benedict test for um, reducing sugars. What are reducing sugars? Reducing sugars are sugars that donate electrons, the electron donors. Okay, in this case the Benedict solution is the reagent. The Benedict solution contains copper sulfate, okay, copper sulfate, copper sulfate. All right, so reducing sugars, what they do is that they donate electrons to copper. Now the copper sulfate, all right, there is plus two plus. All right, so an electron is donated to it, it changes. Of course, this is red or brick red. Is that okay? Yeah, so when electron is donated to it, it changes, all right, to copper 1. No, this is not red. Copper 2 is not red, is blue. Okay, that is why Benedict solution or copper sulfate is blue. So this is blue. Okay, so when electron is donated to it, guys, what do we have? Okay, one electron is given to copper 2, give it one electron. Okay, um, by this reducing sugars, right, it will form copper 1, that's copper plus. Okay, copper plus is brick red or red. Okay, all right, now anytime it's reduced turns to red, but depending on the composition, the quantity of copper, right, one ion present. So remember, the first thing is reagent. The reagent is Benedict solution. The second thing is procedure. So what do you do, guys? In the laboratory, what do you do? So what you do is, first of all, you get a test tube, rinse the test tube, and then you add 2 ml of the test solution. Okay. If a manufacturer claimed that the fruit juice contains reducing sugar, okay. Meanwhile, don't forget that reducing sugar includes all monosaccharides. Okay, all monosaccharides. All monosaccharides. Monosaccharides are the building blocks, right, of polysaccharide. So all monosac, I'll call it monosaccharide. Okay, monosac here plus lactose, all right, and maltose. So lactose and maltose are disaccharide. Is that okay? So we have lactose, they also are reducing sugar. So when we talk about all monosaccharides, we'll talk about glucose, galactose, fructose, ribose, deoxyribose, they're all monosaccharides. But we have disaccharides, which are lactose. Okay, lactose is made up of glucose and galactose. Okay, we have a topic on this, right? So we have maltose. Maltose is glucose and glucose. So they are reducing sugar. So if they are present, all right. Now, I mean, now these are disaccharide, okay? But we'll have glucose and the rest, they are also monosaccharide. So if you what you do is that if you want to check whether reducing sugar, okay, is present in a juice, all right? Fruit juice, let's assume this is fruit juice, all right, contained here. So what you do is you just add 2 ml of the test solution. 2 ml of test solution. 2 ml, same thing at 2 centimeter cube of tea. I'll call it test solution. That is what you want to test. And then to it, you add 2 ml. Okay, just like what? So 2 ml or more, 2 ml or more volume, okay, of Benedict solution. So you add Benedict, 2 ml or more, okay, of Benedict or more. So you add more, okay, it's of Benedict. I'll call Benedict B. Is that okay? Yes, Benedict solution B. Okay, so you add all of that inside this test tube. Okay, mix them, and the next thing is do is that, like just like stated here, okay, Benedict solution and then mix, right, and then heat. The next thing you do is that you heat plus heat. That's all you need to do. That's a procedure, guys. And you wait for the result. So what do? Equal volume of sample being tested and Benedict solution. So let's add 2 ml of the tested sample, add 2 ml of Benedict solution into this test tube, and then put it in a water bath or heat up to 95 degrees Celsius, this degrees Celsius, for 5 minutes. After that, you check for the result. So if reducing sugar is present, whether it is glucose or galactose or fructose or even these disaccharides are present, okay, the blue color will change to green. Remember that Benedict, all right, is Benedict's reagent is blue, okay, so that blue color will change from blue to either green or yellow. This is all, not to. Okay, this is green or yellow or orange or brick red, depending on the concentration, guys. So if the concentration is high, for example, if the concentration is high, all right, it will change to brick red. So brick red shows us the highest concentration. This is highest, all right, highest concentration. Okay, brick red shows highest concentration, all right, highest concentration. Now if it is the lowest concentration, very small, all right, is green. But if it is moderate, between yellow and orange, is that okay? That is if it is present. So if it is present, we're going to have the Benedict solution, which is copper, which has copper 2, which is blue. Okay, remember that when electron is donated to copper 2, it's going to change color. Now it's going to change depending on the concentration of Benedict solution of the reducing sugar, and that of copper 2, all right, is going to change from blue to either green, lowest concentration, or yellow or orange, all right, these are moderate, or highest concentration, brick red. So if that happens, that means reducing sugar is present. But if it doesn't change color, that means it remains blue, reducing sugar is absent. Okay, all right, now knowledge check, guys. All right, now say to estimate the concentration of glucose in an unknown solution, equal volumes of range of known concentration of glucose were each mixed. with the same excess volume Benedict after Benedict solutions. After mixing the solutions, the solutions were placed in a thermostatically controlled water bath at 90 degrees for 3 minutes. Okay, just to heat it. Remember what we did at the beginning, what was said at the beginning. All right. So what happens? The unknown solution was then treated in the same way, and the colors of the known and unknown solutions compared. What is the independent variable in this procedure? Okay. All right. Remember, they said to estimate the concentration of glucose in a known solution, equal volumes of a range of known concentrations of glucose. All right. I want you to pause this video and try to answer this question. Pause the video and answer this question. Okay. Yeah. But guys, remember, if you are conversant with scientific experiment, you know that in every scientific experiment, we have the independent variable. The independent variable is what you are investigating. All right, that is what you change. You alter the independent variable. The dependent variable is what you are measuring. Okay, constant variable is what you keep constant. We call it key variables or controlled variables. So in this experiment, what were we changing? They said to estimate the concentration of glucose in an unknown solution. So we're estimating, that's why we're investigating this. Equal volumes of range of known concentrations of glucose. So we are changing the concentration, we're altering it, but they have the same volume. So that means volume, all right, is the constant variable. Okay, are you following? The volumes of any solution, volumes of the glucose concentration, they must be the same. But what are we changing here is concentration. So concentration is the independent. Independent is what you change, what you vary. Okay, like final colors of solution is the dependent variable. That's what we want to measure. Okay, we use the final colors to determine the concentration. So concentration is measured using final color. So this is independent. All right, this is dependent. Temperature of water bath should be the same for all. That is constant variable or controlled variable. Volume controlled. So control, dependent, that's what we measure. Independent, that's what we change. So the answer is A. Is that okay? All right, let's go to the next question. Now, this is the test for non-reducing sugar. Guys, the only non-reducing sugar we have is sucrose. Okay, so this is when you hear non-reducing sugar, all right, that's the test for sucrose. Don't forget. Test for sucrose is a test for non-reducing sugar. All right. Okay. So sucrose is the non-reducing sugar among the simple sugars. Is non-reducing sugar. Of course, polysaccharides are not. They are not. Of course, regarded as simple sugars. So sucrose consists of glucose. Remember, sucrose is transported. The form of carbohydrate transported in plant is sucrose. So sucrose consists of two monosaccharides, glucose and fructose. Okay, so that means that you see sucrose is non-reducing. It cannot donate electron, okay, to copper 2 ion in Benedict solution. Okay, so of course that is why it is non-reducing. So how do we check whether it is present in a sample? Guys, don't forget, remember, the first thing you do is that you carry out tests for reducing sugar. Please, if you want to do this, do tests for reducing sugar. Why? Because test for reducing sugar is the same as, the result is the same as that of non-reducing sugar. Okay, the result is the same in the sense that they will always change from blue to either, if they are present, either from blue to either green or yellow or orange or brick red. So to be sure that non-reducing sugar is not present, carry out tests for non-reducing sugar, and after that, if the result is negative, then you go ahead to do this. Okay, so now the first thing you do is that you take equal volumes. Okay, just like we did, equal volumes of course, that's the first thing. Equal volumes of the test solution put in test tube, right? yeah, equal volumes, that's 2 ml, all right, of test solution, right? You add to that test tube in the lab, right? You add it an acid, okay, or an enzyme. The reason is that we want to hydrolyze this sucrose. We want to break the bond, the glycosidic bond joining glucose and fructose together. So it needs to be broken. That's why we are adding acid. Is that okay? If acid is not present, you use an enzyme that can hydrolyze. So the reason for adding the acid is to hydrolyze, okay, or the enzyme. So if you check what we have on the slide here, say hydrolysis is followed by Benedict test, okay, for non-resistant sugar. So first of all, hydrolyze and then test. So hydrochloric acid, after you have added the test solution into the test tube in the lab, please, about 2 ml, all right, you now add, all right, of course almost the same, like 2 ml or less of the acid, 2 ml or less of the hydrochloric acid, okay, I'll call it A for acid. You add it to the same test tube here, add it there, okay. So you add 2 ml of test solution, 2 ml of acid A, and then you now heat. So you see what we have here, okay. The hydrochloric acid is catalyst, okay, is added to hydrolyze the sugar in the sample being tested in the ratio of 1:2 respectively and heated in water, both, okay, but for approximately 2 minutes. So by the time you add this, then you now add heat in a water bath, and then that this H is heat, right? You heat the mixture. Why are you heating it? To hydrolyze sucrose. Remember, the first thing you do, you add 2 ml of tea into this, that's the solution. Then you add 2 ml or half of it, okay, to the same test tube, and you apply heat. After 2 minutes, you now take a pinch of sodium hydroxide is added. Why? To make the solution alkaline, because Benedict solution does not work in acidic solution, okay. So acidic pH, so it works in alkaline. So you add the sodium hydroxide to it. So remember, after you have heated for 2 minutes, you can cool it, all right, and then you have to cool it, right, and then you can add your base, that is sodium hydroxide, to neutralize the acid and create alkaline environment, and then you now add your Benedict solution. You add Benedict solution, okay, you add Benedict solution, I'll call it BD, right, Benedict solution BD, and then you now heat again. That's all, plus the last one, heat, okay. So after you have added the Benedict solution, you have to heat again, okay, in water bath, all right, for about 5 minutes. So let's go over it again. So you want to test for non-resistant sugar. What do? Take the test solution, about 2 ml out to test tube. Take about half or equal volume of acid, hydrochloric acid or enzyme, add to this. If you're using acid, you have to, of course, you're going to add the base. But first of all, add the acid with the test solution, then heat. After heating it cool, you add the base, which is sodium chloride, so sodium hydroxide, okay. So you know when to heat. When you add the acid and this, you heat, and then you add the base, and then add Benedict solution, and then heat again and wait for the result. Like I told you, if resistant sugar is present, all right, the blue color of Benedict will change to either green or orange, or yellow or orange or brick red, just like the Benedict test, okay. So after this Benedict test is carried out, so the same result is gotten, okay, all right, the same result. If this is present, it will turn from blue to green or yellow or orange or brick red. But if this is not present, it will remain blue, okay. So knowledge check 2. Now look at knowledge check 2. Steps 1, 2, 3, and 4 are used to test for non-resistant sugars. So what did they put in? Number 1, put 5 cm cube of solution into a test tube, add a few drops of acid, okay. Yeah, why are we adding the acid? For hydrolysis. You want to hydrolyze sucrose if it's present. Then 3, neutralize with alkali RCCMQ of any solution. Now some things are missing here. They say, when is the solution heated or boiled? When is it heated or boiled? Pause this video and try it. When is solution heated or boiled? Pause the video and try it. Okay, so when is it heated? Now what, by the time you add this test solution into the test tube and then put the acid, then you have to heat. Okay, so after 2 you heat, so between 2 and 3 you have to heat. Then when you neutralize and add Benedict solution, okay, you have to heat again. So A says between step 1 and 2 no, between step 2 and 3 yes, after step 4 yes. All right, so B is the answer. C says between steps 2 and 3 yes, but not only, because after you add Benedict solution you have to heat again. All right, it's not after step 4 only, it is between steps 2 and 3 and after step 4. Okay, let's go to the next question. Check, this is knowledge check 3. Let's check what you know. Which flow chart outlined the test for non-reducing sugars is correct? Now look at this. Add alkaline sodium hydrogen carbon to the sample. Is that the first thing you add? No, but pause the video and try it. Pause this video and try it. Okay, look at so A can be the answer. All right, because you don't start with adding alkaline. Now the second one was I don't start with alkaline. So what did you boil with dilute hydrochloric acid? So A and B eliminated, C and D adopted. Let's work them out. So boil sample with dilute hydrochloric acid to hydrolyze sucrose, yes. Then neutralize with sodium hydrogen carbonate, yes. And then add Benedict solution and boil. Good. This C is the answer. But let's check why is D the wrong answer. When you boil sample with dilute hydrochloric acid, neutralize with sodium. Yeah, this is correct. You don't add burette. Burette is not used for testing non-reducing sugar. You use Benedict solution. Okay, so the answer is C. The answer, all right, is C. C is the answer. Okay, all right. Now look at knowledge check 3 to tell knowledge check 4. All right, it says many flowers produce sweet solution called nectar. Wow, very sweet. Bees provided with nectar. So if you give bees nectar, they use enzyme Q to change the nectar into honey. Wow, honey. Give bee nectar, bee will give you honey. Wow. They say after testing a sample of nectar, now test that, okay, for the presence of reducing sugar using standard laboratory reagent. All right, the sample was blue. That means it does not have reducing sugar. Okay. After testing a sample of honey in the same way, the sample was orange. Well, what does it mean? That means it has reducing sugar because there's a color change from blue to orange. Which conclusion about the reaction catalyzed by enzyme Q is consistent with this result? All right, pause this video and try it out. Get me the answer, I'll send you chocolate. Okay, so watch this. Remember, we are told that nectar doesn't have, right, because after they carried out testing for nectar, right, using standard laboratory, it was blue, means no reducing sugar. That means I suspect that nectar had a non-reducing sugar, sucrose. Is that okay? Because sucrose, if you boil it with reducing sugar, so with Benedict solution, it would not change the color, all right, from blue to any other. So this is nectar N, okay? But when we gave nectar to bees, bees converted our nectar, all right, by using this enzyme, enzyme Q, changed nectar to honey. Good, right? And then we'll carry out the same test after testing the sample of honey, right? So what do we have? We have orange, so it showed positive result. That means that this enzyme Q did what to sucrose? It did what to nectar? This is nectar. Nectar contains sucrose. Okay, how do we know? It gave us negative result, no change in color, right? So enzyme Q converts, right, sucrose, nectar to honey, right? Good. So honey showed the presence of what is it called? Reducing sugars. So that means that for the reducing sugars to be formed, we have two reducing sugars present in sucrose is glucose and the fructose. Okay, yeah, fructose. So now what did that enzyme do to sucrose? Okay, let's go to the options. Okay, so options A says condensation. Condensation means joining the two together. Joining these two together as condensation. You know, they didn't join them together. So A and B are wrong, because what the guy did here, what this did, just it broke them down into glucose and fructose. That's hydrolysis. Hydrolysis means breaking down. So C and D are on our distance. So what is the substrate? Sucrose. Because if it were maltose, maltose is a reducing sugar, it would have changed color when the nectar was tested. Okay, so it's sucrose, and the product is fructose and glucose, but it's okay. So that means that our answer is option C, Charlie. All right, let's go to the next test quickly. So the next test says Burette test used to dictate the presence of protein. Yeah, we use it to detect the presence of protein. So equal amount of the sample and Burette solution are added together, giving purple color ever several minutes in the presence of protein, and blue in its absence. Okay, guys, remember, in order not to get confused, remember that the first thing you check out is reagent. So what's the reagent? R represents reagent. The reagent for test of protein. We are testing proteins, right? For peptide bond. So our reagent is burette. Guys, don't forget that. Now burette, all right, is a mixture of copper sulfate. So if you don't see burette on your bench table, look for copper sulfate, right? Look for KOH. A mixture of these two compound, all right, gives us burette. Okay, so what do? This is your test tube. Rinse the test tube, right? Add, what do you add here? You add okay, about 2 ml, right, of test solution, right? 2 ml of tea test solution. Add it to the test tube, and then this is simple. And then add 2 ml of burette. Is that okay? So if burette is not present, you add 1 ml of each of copper sulfate. Is that okay? Because two of them make up, all right, the burette reagent. Of course, I'm talking about P, procedure, all right, and then result, RPR, reagent, procedure. See, this is reagent, R, procedure, and result. So what is our reagent? Burette. What is our procedure? 2 ml of test solution to the test tube, 2 ml of burette into the test tube. So what is our result? Our result is that the color of burette is pale blue. Okay, pale blue, pale blue. So it's going to change from pale blue, very important, pale blue to what? All right, to purple, right, or lilac, or mauve, or violet. Okay, all right. So if this happens, that is positive results. Okay, but if there's no change in color, guys, if this remains the same, right, same means negative result. So if the color doesn't change, that means negative result. Okay, all right. So that's how you know that protein is there. Okay, let's look at emulsion test. Emulsion test is a test for lipids. Okay, the sample is added to 2 cmc. Now 2 cm of ethanol mixed well until it dissolves. Okay, lipids are soluble in ethanol, so ethanol is our reagent here, and water. Okay, ethanol. So what that means is, just like they mentioned, take 2 ml of the test solution, add it to a test tube. Okay, 2 ml, okay, minimum. If you want to, of tea, add it to that, and then add to that 2 ml of ethanol. Do you understand? In the lab. So add 2 ml of what? Ethanol. Ethanol is E. All right, to form a mixture. And then there's another test tube that has water. This test tube has water. Okay, so what you do is that when you have poured 2 ml of when you have mixed 2 ml of test solution and ethanol in a test tube, pour the mixture of this into water. Don't pour water into them, please. The mixture of test solution and ethanol, okay, like we said here, this mixture is then placed into a test tube containing the same amount of water. So that means this water, all right, we have about 2 ml, 4 kg, like 4 ml, all right. And then if lipid is present, what will be the positive result? A milky white emulsion will appear if lipid, are present, all right. But if it's not present, it will form a clear solution, okay, a clear solution. All right, now iodine test for starch quickly. Now this is simplest stage test. Don't forget that iodine is the it is the reagent, RPR, reagent procedure result. So the first thing to ask yourself is, what is the reagent? Iodine, and the color is orange brown. So you just mix add a drop of iodine solution to the solid or liquid substance to be tested. A blue-black color is quickly produced if starch is present. Yeah. So once you mix them, starch, the color will change from orange brown to what? Blue black. But if it's not present, it will remain orange brown. Okay, so put few drops of iodine in a few drops on a white tile. If this is a white tile, so few drops of maybe test solution on the white tile, and then on top of the drops, drop iodine and check the color change. So if starch is present, changes from orange brown, iodine changes from orange brown or brown, okay, orange to blue black. If not, okay. Knowledge check 5. Pause the video and try it. Pause the video. See, the Benedict test and ethanol, the Benedict test and ethanol emulsion test were carried out on a sample of biological molecules. The solution became brick red during Benedict test and cloudy during ethanol. Which molecules did the sample contain? So guys, what's the answer? Okay, if the Benedict gives us, okay, Benedict gives us what? Brick red. What does it show? What does it show? It shows that a residue sugar is present. And if emulsion gives us cloudy or white, it shows that lipid is present. So A says this is a residue sugar and lipid. Yeah, present. Protein is not present. Starch is not present. Non-residue sugar is not present. So the answer is alpha. Okay. So thank you for watching. I want you to look out for part 2 of biological molecule. Okay. Please kindly watch out for the next part and leave your questions at the comment section. Like, subscribe, turn on the notification button on 13 Universal YouTube channel for prompt notification on our new videos. Thank you for watching. Okay, thank you. This is Shedro. Bye for now. See you next episode.